Hello friends, welcome once again in another video of Easy Life Science. Here I will be talking about some important questions, their answers with detailed explanation for SET exam subject Life Science part 13. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon to get the notification of my newly uploaded video. So let's start. Here I am going to continue from part 12. The question: the condition in which the host cell chromosome carries a bacteriophage DNA is known as. Here the question is asked on the lysogeny. In a lysogeny, the viral DNA get integrated into a host cell chromosome and it becomes a prophage, or simply the prophage is a DNA of a virus which is integrated into the host cell chromosome. So answer of this question is a prophage. Then the other option, the temperate phage. The temperate phage is a term given to the virus which cause a lytic cycle or a lysogeny to the host is known as a temperate phage. For example, lambda phage is a temperate phage. Then the option C, lysis. In a lysis, after the infection multiplication in a host cell cytoplasm, the virus gets assembled and number of virus particles are formed. After formation of a number of virus particles, they get released via a lysis of a host cell. In a lysis, cell get burst. Lysogen, it is a name or a term given to the host to which the virus integrates its DNA. So for this question, answer is option B. Then the next question, which one of the following is a cell adhesion molecule? Answer of this is option A, integrin. Integrins are transmembrane receptor that facilitate cell to cell and cell to extracellular matrix adhesion. Most integrins are connected to bundles of actin filaments. So answer of this question is option A. Then looking for the other options, lysine, it is a essential amino acid. Meaning of it means the human body cannot make it, and you have to get the lysine from food or supplements. Then option C, myosin, it is a superfamily of motor proteins best known for their role in muscle contraction and in a wide range of other motility processes in eukaryotes. They are ATP dependent and responsible for actin based motility. And option D, that is a keratin, it is a structural fibrous protein. It is the key structural material making up scales, hairs, nails, feathers, horns, claws, hooves. Or we can say that it is a protein found in epithelial cells. So answer of this question is option A. The next question, junction within the cell which prevent a small molecule from passing in between two cells are termed as. The answer of this is a tight junction. The gap junctions are aggregates of intracellular channels that permits direct cell to cell transfer of ions and small molecules. One gap junction is composed of two protein hexamers called connexons, whereas a tight junction, the tight junction forms the continuous intracellular barrier between epithelial cells, which is required to separate tissue paces and regulate a selective movement of a solute across the epithelium. Adhesions are the bands of a scar-like tissue that forms between two surfaces inside the body and cause them to stick together. Desmosomes, they allow transfer of a water from one cell cytoplasm to another. So, answer for this question, junction within the cell which prevent a small molecule from passing in between two cells is termed as a tight junction. Then the next question, delayed type hypersensitivity is mediated by. Delayed type of hypersensitivity is mediated by T cells. Looking for the another options, antibody are the globular proteins which are synthesized by our immune system for giving a response to a particular antigen. B cells are responsible for production of the antibodies. Then the next question, a small organic molecules which are antigenic but not immunogenic is called as. A small organic molecule which are antigenic but not immunogenic. The answer of this is a hapten. Hapten is a small molecule that stimulates the production of antibody molecules only when it is conjugated to a larger molecule that is a carrier. Many small molecules such as toxins, drugs and hormones are not capable of invoking immune system when injected directly into the animals. They are thus not immunogenic but themselves are also called as a haptens. The other option, that is option B, adjuvant. Adjuvant is a substance that modulates the immune response to a vaccine. Adjuvants may act by a combination of various mechanisms including formation of cytokines, chemokines, recruitment of immune cells, enhancement of antigen uptake and presentation, and promoting antigen transport to draining lymph nodes. Option D, that is a epitope. An epitope or antigenic determinant is a group of amino acids or other chemical groups exposed on the surface of a molecule. Frequently, it is a protein which can generate an antigenic response and binds antibody, or simply a epitope is a small site on an antigen to which a complementary antibody may specifically bind. So the answer of this question is a hapten. Then the next question, inhibin form a Sertoli cell of a testis selectively inhibits. The inhibin is a protein secreted by a Sertoli cells of a testis in a man and by a granulosa cell in a woman. It inhibits the synthesis and release of the follicle stimulating hormone in the pituitary gland. So it selectively inhibits a FSH, that is a follicle stimulating hormone. Then the next question, during fertilization, polyspermy is prevented by. The polyspermy means the fertilization of an egg by more than one sperm. Diploid organisms normally contains 2 copies of each chromosome, one from each parent. The cell resulting from polyspermy contains 3 or more copies of each chromosome, one from egg and one from multiple sperms. This may occur because sperms are too efficient at reaching and fertilizing the eggs due to a selective pressure of a sperm computation. And to prevent a polyspermy, the zona pellucida carry out a important role. It is a structure that surrounds the mammalian eggs and it becomes impermeable upon a fertilization and it prevents the entry of further sperms. In a fast blocking of a polyspermy, it is achieved by the changing the electric potential of a egg plasma membrane, and this membrane provides a selective barrier between egg cytoplasm and outside the environment. Polyspermy is also prevented by a acrosome reaction, the release of hydrolytic enzyme which softens the zona pellucida. The zona pellucida is a jelly coat A cortical reaction, it hardens the jelly coat, and post fertilization event are prevented like a polyspermy. So during fertilization, polyspermy is prevented by a cortical granules or a cortical reaction. Now the other option, that is a phytolene membrane. The phytolene membrane is a structure surrounding the outer surface of a plasma membrane of an egg. The hyaluronide enzyme, this enzyme, hyaluronide enzyme, depolymerizes the hyaluronic acid polymers in the intercellular cement that binds the ovarian granulosa cells together. The next question, the very first event of a mammalian fertilization. In a fertilization, a sperm nucleus fuses with a egg cell nucleus and forms a zygote. Number of events occurs in a fertilization. First, that is a sperm capacitation. Second, sperm zona pellucida binding. Third, acrosome reaction. Fourth, penetration of a zona pellucida. Fifth, sperm oocyte binding. And sixth, egg activation and cortical reaction. And seventh, zona reaction. Eighth, post fertilization events. These are different steps which occurs in a fertilization. The very first event in a fertilization is the capacitation. I have one image, have a look on it. Firstly, the structure of a sperm. The sperm is with a head, neck and tail. In a head, there is a nucleus of a sperm. To the top of head, there is the acrosome. At a middle, there is a mitochondria of a sperm, and this is a tail. This is a overall structure of a sperm, and this is a egg cell. The first event, the sperm approaches to the egg. Second, the sperm's acrosomal enzyme digests the egg's jelly coat. Third event, the proteins on a sperm head bind to a receptor. Here is a receptor on an egg known as a receptor protein molecule, to which the proteins on the head of a sperm get binded. In a fourth event, plasma membrane of a sperm get fused with a plasma membrane of an egg cell, and after fusing, the sperm release the nucleus from a head to the cytoplasm of an egg cell. This is a nucleus of a sperm, and the nucleus of a sperm get fused to a egg nucleus and forms a zygotic nucleus, and this is a fertilization. So question is asked, the very first event of a mammalian fertilization is the capacitation. The next homobox sequences are. Homobox sequences is a DNA sequence around 180 base pairs that regulates the anatomical features in early stages of embryonic development, and these are regulated by a gene known as HOX genes. HOX genes. So answer of this question is option B, junk DNA sequences. These are not any kind of integration site for viruses or conserved sequence. So option B is the answer of this. Then moving to the next question. Which among the following is not substrate for Rubisco? Rubisco, that is ribulose 1,5 biphosphate carboxylase. This is a enzyme which is present in a large amount in universe, and this enzyme carry out a photosynthesis. The substrate for this enzyme are ribulose 1,5 bisphosphate. This is a main substrate ribulose 1,5 bisphosphate, a 5 carbon containing compound having a 2 phosphate molecules, and CO2 and O2. These three are the substrate for the Rubisco. When the Rubisco molecule having a substrate ribulose 1,5 bisphosphate and CO2, the enzyme carry out a photosynthesis and forms 2 molecules of 3 phosphoglycerate, and this 3 phosphoglycerate forms a reverse of a EMP pathway and forms a glucose molecule. But when the ribulose 1,5 bisphosphate and O2 is there, the enzyme carry out a photorespiration and 1 molecule of 3 phosphoglycerate and 1 molecule of 2 phosphoglycolate are formed. This is known as a photorespiration. CO2, O2 and ribulose 1,5 bisphosphate, these three are the substrate for Rubisco, but glyceraldehyde 3-phosphate is not a substrate, it is a first stable compound in a photosynthesis. So answer of this question is option B. The next question, which one of the following is not correct about a glyabulate cycle? Glyabulate cycle is a variation of a TCA cycle. It is an anabolic pathway occurring in plants, animals, fungi and protista. The glyabulate cycle centers on the conversion of acyl coenzyme A to succinate for synthesis of carbohydrates. In the microorganism, the glyabulate cycle allows cell to use 2 carbon containing compounds such as acetate to satisfy cellular carbon requirements when the simple sugars such as glucose or fructose are not available. And this cycle is absent in the animals. So it is a variant of tricarboxyl cycle, or it is a variant of TCA cycle. It results in the formation of a succinic acid from acyl coenzyme A. It takes place in glyoxysomes, a special kind of cell organelles where this cycle occurs. And the option D, it says that it occurs in all eukaryotes. It is wrong. It occurs in plants, fungi and protista, not in animals. So this option is incorrect. It does not occur in animals. The next question, which of the following is non-nitrogen fixer? Rhizobium, Frankia, Physia fava and Azosporidium, these are the options. Rhizobium, it is a genus of gram-negative soil bacterium that fixes a nitrogen. This species fixes a nitrogen by endosymbiotically with association to dicotyledonous plants or root nodules of these plants. They are symbiotic nitrogen fixing bacteria, gram-negative in nature, present in a soil. In a nitrogen fiction, bacteria colonize plant cells within a root nodules where they convert atmospheric nitrogen into ammonia using an enzyme nitrogenase. Option B, that is Frankia. The Frankia fixes atmospheric nitrogen in a symbiotic association with non-legume actinorhizoid plants, for example Alnus, or it fixes a nitrogen in free living states. Then the option C, Physia fava. The Physia fava, it is also known as a broad bean or a fava bean. It is a species of a legume family Fabaceae, widely cultivated for its edible seeds. And this plant does not have any ability to fix a nitrogen. Then the option D, Azospirillum. It is the free living nitrogen fixing bacterium associated with grasses. So here the question is asked non-nitrogen fixture. All of the three, Rhizobium, Frankia and Azospirillum, are nitrogen fixers. But the Physia fava, it is not a nitrogen fixer. Then the next question, plasmolysis occurs when a cell is placed in. Plasmolysis is a process in which cells lose water when placed in a hypertonic solution. It causes a contraction or a shrinking of a plasma membrane away from the cell wall. It is a reversible process and cell can get back to a normal when placed in a hypotonic solution. The isotonic solution and a solution having a exactly equal salt concentration to that of the cells. In a plasmapheresis, when the cells are placed in a hypotonic solution, the water is taken inside the cell cytoplasm and cell get boiled, known as a plasmapheresis. So here question is asked the plasmolysis, and this occurs when a cell is placed in a hypertonic solution, that is option B. The next question, brown sugar and a heroin are the secondary metabolic product of which of the following plants? The answer of it is a Papaver somniferum, or it is known as a poppy. Poppy is a routine name for this plant, Papaver somniferum. The heroin or a brown sugar. Brown sugar is an adulterated form of a heroin. It is also called as a smack, junk, skag and chaw. These are the regional terms: smack, junk, sag and chaw. It is a opioid derived from a morphine extracted from a poppy plant. Poppy means a Papaver somniferum. So answer of this question is a Papaver somniferum. Looking for the other options, Conium maculatum. The Conium maculatum, known as a hemlock. It is a biennial herbaceous flowering plant in a carrot family, Apiaceae. Extract of a Conium maculatum is used as a traditional medicine for a cervix carcinoma. Option C, Claviceps purpura. This Claviceps purpura is also known as a ergot fungi. It is a fungal parasite of cereals and grasses that produces toxic alkaloids. Option D is Rauwolfia serpentina. This is a plant is a source for Rauwolfia. This is a alkaloid, and this Rauwolfia alkaloid is used to treat a high blood pressure, that is a hypertension. So answer of this question is a Papaver somniferum, that is a poppy. The next question, the amino acid which acts as an osmolyte and accumulates high during osmotic trace. The proline is answer of this. Proline get accumulated at time of high osmotic trace. Then the next question, focal length of eye lens is controlled by. It is controlled by a ciliary muscles. The ciliary body is a circular structure that is extension of a iris, the colored part of eye. The focal length of eye is controlled by a ciliary muscles. The cornea, transparent part of the eye that covers the iris and a pupil and allow light to enter inside. Vitreous humor. Vitreous humor, it is a clear gel that fills the space between the lens and a retina of eyeball. So option B is the answer of this. Then the next question, zymogen cells of gastric gland secretes. Zymogenic cells of a gastric gland secrete a pepsinogen. Pepsinogen is secreted by a gastric chief cells as a proenzyme and then converted to a pepsin. So option A is the answer of it. Looking for other options, chymotrypsins. The chymotrypsin is a digestive enzyme and a component of pancreatic juice acting in a duodenum where it performs a proteolysis, that is a breakdown of proteins and polypeptides. It cleaves a peptide amide bond from a N-terminal. Pepsin is a stomach enzyme that serves to digest proteins found in ingested food. Gastric chief cells secrete a pepsin as an inactive zymogen called a pepsinogen. The parietal cells within the stomach secrete a HCl, and that HCl lowers the pH, and so that the pepsinogen get activated to a active pepsin. So option A is the answer of this question. Then the next question, which one of the following part does not constitute a part of a single urinophallus tubule? The loop of Henle. It is a long U-shaped portion of a uriniferous tubule that conducts a urine within each nephron of the kidney. The function of a loop of Henle is in a recovery of water and a sodium chloride. Bowman's capsule, it is a part of nephron that forms a cup-like sac surrounding the glomerulus. Bowman capsule encloses a pace known as a Bowman's pace. It is a contagious with a proximal convoluted tubule. So, Henle, loop of Henle, Bowman's capsule and glomerulus, these all are a part of a single UT or a single uriniferous tubule, but the collecting duct is not a part of a single uriniferous tubule. Option C is the answer of this question. Then next question, Cushing's disease is related to. The Cushing's disease occurs when the pituitary gland makes too much of the adrenocorticotropic hormone, or simply we can say that it is a hyperadrenalism. Most common cause of a Cushing syndrome is the taking too much glucocorticoids or corticosteroids. These are the hormones, and these are present in medications such as prednisone, dexamethasone and prednisolone. So answer of this question is a hyperadrenalism, option A. The next question, function of a tracheal hairs is 2. Tracheal hairs, the innermost layer of a trachea consists of ciliated pseudostratified columnar epithelium cells. On the surface of columnar cells, long hair-like cilia beats together to push mucus away from the lungs. It is expelled out during a coughing. So read the options carefully. It passes a mucus outside. Option D is the answer of it. By mistake it is written as a B. Option D is the answer. The function of a tracheal hairs to pass mucus out. The next question, functional unit of a gene that specify synthesis of one polypeptide. One polypeptide is synthesized or coded by one cistron. One cistron, one polypeptide. Cistron is a minimal genetic unit which is responsible for formation of one polypeptide. Codon, the codon is read on a mRNA. It is triplet in nature, having 3 nucleotides and gives a synthesis of one amino acids. Muton is a minimal genetic unit which is responsible for undergoing a mutation. Recon is a minimal genetic unit which is responsible for a recombination. So answer of this question is a cistron. It is a gene or a minimal genetic unit which is responsible for synthesis of one polypeptide. So next question, if a mutation in a gene responsible for particular autosomal dominant disorder has 80% penetrance, then how many of the persons with the particular mutation will develop the disease? The penetrance is 80%, so the possibility of a person to have a particular mutation will be 80%. Answer C is the answer of it. The next question, which of the following is a horizontal transfer of a gene? Transfer of a DNA from parent to offspring, not it is a vertical one. Transfer of RNA from DNA template, it is a vertical. Translation, it is not a gene transfer but a transmission of a genetic material from one natural organism to another. It is a horizontal gene transfer process. Transformation, conjugation and transduction. In a transformation, 2 cells are there, one is acting as a donor and another acting as a receptor. The donor cell, its cell-free DNA is accepted by a receptor 1 and forms a endogenous and recombinant cells are produced. In a conjugation, cell to cell contact is there in between donor and recipient, and because of a F plasmid or a fertility factor, they becomes a recombinant. And in a transduction, through the help of bacteriophage, there is a transfer of a genetic material from donor to a recipient. So option D is the answer of this question. Then next question, recombination occurs between, it is a question related to a cell division. It occurs in between the chromatids of a homologous chromosomes. Option B is the answer of it. Then the next question, hemophilia is a X-linked recessive disease. Hemophilia is a disorder in which blood does not clot normally. When a blood cannot clot properly, excessive bleeding occurs after an injury or a damage. Two types of hemophilia are there, hemophilia A and hemophilia B. The hemophilia A, which occurs due to a low amount of a clotting factor 8, whereas a hemophilia B is due to a low amount of a clotting factor 9. So this is a X-linked recessive disease. Option A is the answer of this question. The next question, which one of the following is not a genetic marker? A sequence of a DNA which is known, a sequence of a DNA with a known physical location on a chromosome is known as a genetic marker. Genetic markers are the genes that are close to each other on a chromosome and tends to be inherited together. Most common types of genetic markers are identified by different techniques like RFLP stand for restriction fragment length polymorphism, that is RFLP, RAPD random amplification of polymorphic DNA, and by using isozyme or alloenzyme techniques, AFLP amplified fragment length polymorphism, SSR simple sequence repeats. So SSR, AFLP, RAPD, RFLP, these all are the technique for identification of genetic markers, but the SINE, that is a shiny, these are not any genetic marker. These are short interposed nuclear elements which are non-autonomous, non-coding transposable elements. So option B is the answer of it. So next question, ecosystem with highest productivity. In a terrestrial ecosystem, tropical rainforest show the highest productivity, and in an aquatic ecosystem, the coral reef shows a highest productivity. So option B is the answer of it. Then the next question, which of the following is not a positive interaction? The question is asked on association in between the two organisms. First option, symbiosis. A symbiotic association, it is a association between two different species where the both partners get benefited. Symbiosis can be obligatory, which means that one or more symbiotant depends on each other for survival, or it can be a facultative or optional when they can live independently. Obligate symbiotic relationship, for example, in a lichen, fungal partner cannot live their own. They depends on their algal partner. So it is an obligate symbiotic relationship in a lichen. In a facultative symbiotic relationship, the algal or a cyanobacterial symbiotant in a lichen such as Trintifolia can generally live independently. Commensalism, it is an association between two living organisms where one benefited and other is not benefited or harmed. Example, hermit crabs using gastropod cells to protect their bodies. Mutualism, it is also a relationship like a symbiosis. When two different species work together, each benefiting from their relationship. Example, the bird oxpecker and a zebra. Parasitism, in a parasitic relationship, the parasite benefits while the host is harmed. In a amensalism, it is an interaction where one species is harmed or killed by other and one is unaffected by other. So all of the three, that is symbiosis, commensalism and mutualism, are positive interactions. The partners get benefited, but in amensalism, one get killed and harmed and other remains unaffected. So this is a negative type of interaction. So answer for this question is option D. The next question, the rate of energy storage at consumer level is known as. The productivity refers to the rate of generation of a biomass in ecosystem. The secondary productivity means the production of organic matter by a consumer. Production of organic matter by the producer is known as the primary productivity. The NPP, net primary productivity can be defined as the rate of accumulation of a biomass or a energy. The energy utilized during a metabolic processes such as respiration is excluded from the gross productivity. Or we can say that NPP is equal to GPP minus RA, that is respiration. The GPP is the amount of organic carbon produced by the plant that is not consumed by their own respiration. Or we can say that it is a rate at which solar energy is captured in sugar molecules during the photosynthesis. It is a energy captured per unit area per unit time, that is a GPP. So here the question is asked, the rate of energy storage at a consumer level is known as a secondary productivity. Option C is the answer of it. So thank you for watching me. If you like my video, please subscribe my channel Easy Life Science, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. Thank you once again.